Okay, why don't we get started? So somebody asked me this morning about the readings and that she was going over the chapter and there were more terms and different topics in the chapters that I assigned than I had gone over the class. That's a really good question. So the answer is that you are responsible for all of the topics and the terms that I go over in lecture. So you use the book as a supplement to get a little bit more background in those topics that I go over. If you read in the book something that I didn't mention at all, you are not responsible for that for the exam, okay? So it makes it a little bit easier. The book is very comprehensive. We're going over a good subset, but it is a subset of the book. So you're only responsible for the things in lecture, which is why it's so important for you to be in lecture for the semester. I changed all of the handouts to PDF format. Are people now able to download them? Any problems going? I see some nods. Okay, great. Now, very, very important correction, and we can thank your colleague back here. What is your name? Samantha. So Samantha noticed that in the book, I had actually reversed the functions of the sympathetic and parasympathetic nervous system. This is the correct correspondence. So the sympathetic nervous system is fight or flight, and parasympathetic nervous system is rest and digest. I apologize, the terminology was just flipped on my slide. On the slide that is now on the slides that are now on Blackboard, this is correct, okay? I corrected it. So now go back and if you want to use these slides, use the version on right now. But the correct correspondence is sympathetic nervous system is fight or flight. That is the response to terms like colonoscopy, pop quiz, that kind of stuff. Parasympathetic lets you relax, rest and digest the weekend nervous system. Okay, so important correction there. Third, as you'll notice if you download the syllabus, this lecture is a lecture where I'm introducing for you our first homework of the semester. This is a media report, and all of this information is on a handout in Blackboard. This first homework is designed to, again, kind of emphasize how much neuroscience is in the popular literature. So what I'd like you to do is go out and find a neuroscience-related article in the popular press. I don't care when it is, it could be old, it could be new. Just find something that interests you, okay? And be careful, I want this to be a neuroscience-related topic, not just a psychology or behavioral related topic. So having to do with action potentials or anatomy or physiology, but not just behavior. And if you're not clear, then ask the TAs or myself, and we'll be happy to steer you in the right direction. You have some time. It's due on October 10th. And what we're going to ask you to do is summarize the main point of the article, describe the study that was done, identify the hypothesis. You went over hypothesis in this past week's lab, the main conclusions, and whether you were convinced or not, and whether there was any information that you would have liked to know more about this article. But the main point is for you to get out there, read a little bit about what's being talked about right now in neuroscience. And this will be written up in a 1.5 maximum double-spaced paper. And when you turn it in to your TAs on the week of October 10th in your lab section, you should include for them either a copy of the paper or a URL to the article, so they'll have access to that as well. Okay, so that is your homework assignment, homework number 1 of 3 of this course. Okay, so let's jump in to lecture for neural communication. This is kind of a 2-part lecture. There are 2 major aspects of neural communication: electrical properties and chemical properties. And we're going to focus on the electrical properties today. I'll say that this is the core of the conceptual kind of big concepts you're going to need to know for this first midterm exam coming up. It's not for a while, but this is a really important lecture for that exam. Our first exam is I can't find it right now, but the 5th, thank you. Okay, so first exam is on the 5th. So again, I encouraged you last Wednesday to read the book. I would also encourage you to read this chapter, I believe it's chapter 3 in the book, for next lecture as well, as we move on from electrical properties to chemical properties. Okay, so first, what makes a neuron so special? We talked about particular aspects that made neurons unique from other cells. What are some of those things that made neurons unique? Axons? Yes. They have axons. Any other specialized morphology? Dendrites, very good. What about other characteristics of the neurons that are special from, say, a liver cell? Yes? They can communicate, perfect. Okay, structural differences, axons, dendrites, specialized membranes, and functionally, they communicate with each other via electrical and chemical signals. So let's talk about what's the difference between electrical and chemical communication. So there are these two types of communication that happen in neurons. Electrical communication happens within the neuron, okay? From this point all the way down to the axon, we're talking about electrical communication. That's what we're going to be focusing on today. When the neuron then communicates with the next neuron in the chain, we move to chemical communication at the level of the synapse that we're illustrating here. So chemical communication happens between neurons transmitting at the synapse. Okay, so that's actually next lecture. Today we're going to focus on electrical communication within the neuron. That's our goal for today. Okay, so why is this so important? Really, this particular lecture and this topic is so important because electrical signals are really the vocabulary of the entire nervous system. Einstein thought of his theory of relativity because of these kinds of signals. Leonardo da Vinci created his art and conceptualized his art because of this. Galileo came up with all of his major principles because of exactly what we're going to be studying today, the patterns of electrical activity. Okay, so what we're going to start with is what happens and what are the electrical forces of a neuron just hanging out, a neuron at rest. Okay, so a neuron at rest can be considered a balance of electrical chemical forces, and we're going to talk about exactly what that means. First, I have to introduce several terms that some of you may be familiar with. Ions are electrically charged molecules, and there's two different categories. Anions are negatively charged, and cations are positively charged ions. So we'll be talking about, for example, potassium, K, the symbol K. Potassium is a cation. It's positively charged. Sodium, Na is its signal, we'll be talking a lot about today, is also a cation. These different ions, including potassium and sodium, K and Na, are dissolved in intracellular fluid that's separated from the extracellular fluid by a cell membrane. So these ions can be found within the cell, and the fluid inside the cell is called intracellular fluid, and it's separated by that cell membrane from the extracellular space. Okay? So there's differences in the electrical signal on the inside versus the outside of the cell. And this first section, what we're going to do is figure out why that is. What are the forces that are causing the inside to be different from the outside? So let's measure. Let's do a little thought experiment here. You have your happy little neuron in a dish, and you have electrodes to be able to measure the potential across 2 points. This is the ground electrode or reference electrode here in black, and the red is the recording electrode. So what this measures is whether there's any electrical differences in cations or anions, positive or negative charges between these 2 points. Right now, this electrode is in the extracellular space, and the reference is in the extracellular space. Here we blow this up. You can see the electrode tip, this red tip here, is on the outside of the cell, just like the reference electrode. So it's in the same exact extracellular medium. There's no difference. It's exactly the same. There's no difference between these two electrodes. And what you see on the oscilloscope after you put it through an amplifier is zero difference between these two electrodes. But now we're going to actually impale this happy little neuron here. And here you see the electrode piercing through the membrane. And now the tip of the electrode, where we're measuring here, is on the inside of the axon, and the reference is on the outside. What is the difference there? Well, there, now once the electrode goes in, you see this big negative jump of the oscilloscope, where telling us that the inside of the axon is negative relative to the outside of the axon. A lot more negative charge on the inside. And so we're going to ask, why is it negative? What are the forces and the physical properties that make the inside of the axon negative relative to the outside? Simple question, okay? So first, what is the resting membrane potential? That is, what is the charge of the neuron at rest inside? And the resting membrane potential of a neuron is between -50 to -80 millivolts in general. In our example here, it was -60, kind of the average. And it shows, as we said, a negative polarity of the cell's interior relative to the exterior space. But the key question that we're going to address is why is the resting membrane potential negative relative to the outside? And there's two key features that we're going to consider to answer this question, why. One is specialization of the membrane. There's something about the cell membrane that is making this difference, allowing this difference to happen, the negativity on the inside relative to the outside. And the second are several forces that we're going to talk about, two main forces that we're going to talk about. So cell membrane and several forces. Okay, so let's talk about the cell membrane first. The cell membrane, as many of you will know from your high school biology, is a lipid bilayer with two layers of lipid molecules. It's a nice, a strong barrier. But what you need to know about are channels. Channels exist within the lipid bilayer. These channels are proteins, they're little holes. So if the cell membrane, the lipid bilayer, is a wall, sometimes you have a little hole, or let's just call it a little window, called ion channels. These are proteins that form kind of a hole through the entire lipid membrane that connects the extracellular space to the intracellular space. They span the membrane and allow particular ions to pass. And depending on the kind of ion channel, it allows particular ion channels to pass. Now, let me tell you about different categories of ion channels. We're going to be talking today about several different kinds. Ion channels that are just kind of open all the time. They just do the same thing all the time. Doesn't matter what's going on. They're just open to particular kinds of particular kinds of ions, letting them through. But there are other specialized ion channels called gated ion channels. And these ion channels open and close in response to a voltage change. So at a particular voltage, they may be closed, and they only open if the voltage goes to a particular level, okay? So it's much more controlled, voltage-gated ion channels. There are also chemically gated ion channels. These gated ion channels only open if there's a particular chemical in the environment. And the third example is mechanically gated ion channels. We're going to get to that when we get to the somatosensory system of the body, because these ion channels that are found right on your fingertips, for example, will open with physical force. you press your hand, and these ion channels open, and that allows that action potentials or signals, I should say at this point, to then be propagated. Just physical movement can open some of the gated ion channels, okay? But for today's lecture, we're going to focus on two types of ion channels. The kind of vanilla, I'll call, ion channels that are just open all the time to particular ions with not a lot of specialization. And the second type we'll talk about is voltage-gated ion channels. We're going to get to that towards the end of the lecture. Okay, but just know that these ion channels are contributing to the negative potential that you see on the inside, and I'll tell you exactly how. Now, for the resting membrane potential, some of these ion channels are open all the time. These are the vanilla ion channels that I talked about, and some of them, many of them that are found on the neuron cell membrane allow only potassium ions, these potassium cations, the positive ions, to cross, okay? We say that the neuron and the neuron cell membrane shows a selective permeability to potassium. It can enter or leave the cell freely. But what makes it enter or leave the cell? I mean, what are the forces that make it decide, oh, I'm going to go in right now, oh, I'm going to go back out? The thing that makes it contributes to whether potassium is moving in or out of the cell has to do with two forces that drive ion movement. These are very important to understand. The first is something that you should all understand. It is the force of diffusion. Diffusion causes ions to flow from areas of high concentration to areas of low concentration along what's called their concentration gradient. Simple diffusion. If you have no barriers there and there's high concentrations, it's simply going to flow down. How do we know this? We see this every single day with diffusion. If this is potassium, and this is extracellular space, I'm putting potassium in, and there's a huge concentration of the red potassium at the top. I let it diffuse out with a little bit of help from my knife, and it's simple diffusion. All those potassium ions now get distributed, and you can drink your drink. It's exactly the same thing as this. Simple diffusion. Things go, these ions that I just poured in, diffuse from areas of high concentration to low concentration, just like you see in drinks that you make from Crystal Light or whatever you're going to do. Simple diffusion. The second force is called electrostatic pressure. Oh, sorry. Let me go and just show you. This is what I just demonstrated. Diffusion. You're putting in these blue molecules, and eventually it goes. They diffuse equally in this cup of water here. Now, before I go to the second force, let me just introduce the idea that there could be a membrane in this cup, and it could be semi-permeable. So I could put really big ions and real little ions, and only the little ions go through this semi-permeable membrane. So the blue little ions will then diffuse and equally spread between these two parts, but the red are too big to go through, so the red will be concentrated here. Okay? So this is the introduction of the idea of the cell membrane. The cell membrane is there, but it's only permeable to certain things. One of the things it's permeable is to this potassium that I just put in there. The second major force driving ion movement is what's called electrostatic pressure. And that causes ions to flow towards oppositely charged areas. Everybody knows this by playing with those little trains that have little magnets on the end, and you put all your little trains together. And everybody played with the fact that sometimes if you put two trains together, they wouldn't go together, right? Why is that? The same charge. So if you have the two magnets with the same charge, they won't go together. It's only the positive and the negative charge of those little magnets on your little train that go together. In fact, they're really attracted together. Same kind of idea with just on a teeny tiny level, at the level of the neuron. We are talking about electrostatic pressure that causes ions to flow. If you're positive, you flow towards negative. If you're negative, you flow towards positive. If there's too much positive over there and you're positive, you're not going to go over there because there's too much positive over there. And that's shown right here. So for example, if you're a potassium ion, now what charge was potassium again? Positive. So it would be attracted towards what? A negative charge. Great. Okay. So what's negative in the cell? Very important to understand. Within the cell, there are lots of negatively charged proteins inside the cell. They just are there. They're born there. They live there. There's lots of proteins inside the cell. It has to do with, all the machinery that's in there, and they happen to be negatively charged. So the inside of the cell starts out negative because of the presence of these negatively charged proteins. And that tends to make the inside of the cell negative. The other mechanism that you need to know about, that is, I'm going to introduce it now, but it's more important towards the end of the lecture when I get to action potentials. That is the presence of something called the sodium-potassium pump. And what I'll say here is that it does help maintain a resting membrane potential. But at rest, when nothing else is happening, it's not doing much. So let me tell you what it does, and eventually by the end we'll come back to it and we'll see exactly what this does to help maintain resting membrane potential. What the sodium-potassium pump does is it's a specialized pump. It's an ion channel, but it's special because each time it works, it pumps 3 sodium ions, another positively charged ion, out of the cell for every 2 potassium ions it pumps in. This potassium pump uses energy, a lot of ATP. I'm not going to go over all that. It's just energy. In fact, the presence of the sodium-potassium pump in the membrane of neurons is one of the biggest energy users of the cell. Okay? So just know sodium-potassium pump pumps 3 sodium ions in for every 2 potassium ions it pumps out, but it's not doing that much at rest when nothing is happening. Okay, so here's a picture. Let's try and visualize this. Here we have let's say this is a neuron with its bilipid layer cell membrane here. Here we have these big black negatively charged proteins. The big black dots are proteins. And here we have illustrated our sodium-potassium pump pumping out 3 sodium ions for every 2 potassium ions pumped in. Okay? So that exists at the cell membrane. Now, at rest, potassium ions move into, so remember, I told you that within the cell membrane, there are these vanilla ion channels that are open to potassium. Potassium can do whatever it wants, okay? So I'm potassium. I'm out here. I have a free window into the cell. Am I going to go in or am I not going to go in? You're going to go in. Why am I going to go in? Because it's negatively charged, and that's exactly what happens. So at rest, potassium ions move in to the negative interior of the cell because of electrostatic pressure that we talked about. And as potassium ions build up inside the cell, they also diffuse out through the membrane along the concentration gradient. So they go in, and then maybe there's too many that go in because they all got attracted, and then they go back out, and they just come to what's called an equilibrium. Certain number of them go inside. There's more inside than outside because inside the cell is more negative. And you have this free window of potassium channels that let these potassium ions do whatever they want. Okay? Here they are. Here are the potassium ions. They are being electrostatically pulled into the cell because the negative environment here because of the proteins. And they're going in. There's a lot of them in here, fewer out here. And they reach a certain equilibrium. So, you know, they can't all go in or it would be too positive and there's nothing forcing them in. But here, look at the sodium. Sodium has no free entrance. It's a lot on the outside, very little or none on the inside. Okay, so that's a big difference between potassium, open channel inside, gets attracted because of the negative ions, and sodium. No way inside. It's trapped on the inside, trying to get in, can't get in at rest. Okay, so I talked about equilibrium, where potassium is on the inside of the cell balanced with the negative proteins. And we say that potassium reaches equilibrium when the movement out of the cell is balanced by the movement in. Nothing else is happening. We're at a happy medium. And this level corresponds to the resting membrane potential of about negative 60 millivolts. Okay? So these are some of the major forces that are resulting in what we already did. We already showed you what this is. This is the electrode sticking inside the neuron, and we're seeing a negative 60 millivolt potential, mainly because of the negative proteins, but it's balanced off a little bit because the potassium is going in. Why is the potassium going in? Because it has these free window into the cell, the potassium ion that's, the potassium channel that's opened all the time. And poor sodium is still on the outside, can't get in. Okay, so one important equation that I want you to know at least the name of, you don't have to know all the math behind it, but the Nernst equation is a very important equation in neuroscience because it predicts the voltage needed to counterbalance the diffusion force pushing an ion across a membrane. So the Nernst equation is simply useful for calculating how much force you need to counterbalance the diffusion of an ion into the cell, and it helps you predict at what potential, negative potential or positive potential, that ion comes to equilibrium from. So this prediction, as I said, is the resting membrane potential for that ion, the Nernst equation. Just know that. So the Nernst equation will predict that the resting membrane potential for potassium is negative 60. Okay, so let's review. We started with the question, what makes that inside of the neuron negative? Negative 60. How come it's negative 60? And we talked about the major factors underlying the resting membrane potential. One, there are intracellular proteins in there. They just live there and they are negative. Okay? That's one major factor. Second, there is a sodium-potassium pump there, even though we said that it's not doing a whole lot at rest. We're going to come back to that, but that's an important feature. Major feature, permeability of the cell membrane to potassium. Just open channel, no holds barred, just potassium can go wherever it wants to through these channels, but also dependent on forces like diffusion and electrostatic forces. Also, lack of permeability of this neuron, of this membrane to sodium. Diffusion, as I said, and electrostatic forces. All of these things are resulting in the negative potential of negative 60 inside the cell relative to outside the cell. Okay, so these are all the factors and the forces that you need to understand. We're going to ask you why the membrane potential is negative 60, or we're going to ask you what diffusion has to do with the negative membrane potential, or what electrostatic forces have to do, and how they contributed to the negative potential. So that's the level that we want you to understand this resting membrane potential. Explain why it's negative 60 inside the cell. Okay, so let's do a little test. So I'm going to be revealing these. So are there more proteins inside the cell or outside the cell? Inside. Many proteins inside versus outside. What about sodium? Is there a lot inside or a little inside? Little. Very good. Few, little inside. What about potassium? Inside a lot, many, or few? Many. And we didn't talk about chloride or calcium. You don't have to know that. It just happens to be the largest proportion of these ions that we're going to focus on is potassium. Potassium's the only one that's a lot on the inside. All these others are on the outside. Okay, and so you can now look at this figure in the book and really understand and be able to predict where these are. You'll see a lot of the big black proteins on the inside, very few on the outside. You'll see a lot of potassium on the inside, the orange dots, very little on the outside. But what you see a lot of on the outside is the blue sodium, the green chloride, and the white calcium. Chloride and calcium, don't worry about that. But you do need to know and be able to reproduce the sodium, potassium, and proteins inside versus outside. Okay, great. Now, I just described for you in 25 minutes something that it took years and years to identify and figure out. And this was actually Nobel Prize winning work. Nobel Prize in 1963, work done in the 1950s by Hodgkin and Huxley. And first what they did is they measured, they were the first to actually directly measure the inside membrane potential of a neuron. That was actually very, very difficult to do. They had calculated what it should be based on the Nernst equation. They predicted that it would be negative and probably about negative 60. But at that time, it was very difficult to impale neurons. And their strategy was to find the biggest honkin' neuron that they could find and then stick an electrode in, and then they could get measurements, clear measurements. And what they used was a giant squid axon. I'm not talking about these giant squids that are like 3 men length long that you get from the deep ocean. These are pretty darn big squids, and they just have the name giant squid. But let's look, just to give you a feeling for what they did and how they did this. We are going to look at a quick movie here. Okay, squid dissection. Okay, great.
Professor J.Z. Young began his detailed studies of the squid's nervous system in 1933.
So there's a giant squid.
Opening the mantle cavity in the mid-ventral line exposes the squid's true body wall, through which the viscera can be seen. The muscular siphon itself, and on each side, close to the inhalant openings of the mantle cavity, a large ganglion. This is called the stellate ganglion, and when the gill is removed, the reason is clear. Numerous nerves just below the skin radiate from it in a star-like pattern.
This is a ganglion.
Professor Young showed that each stellate ganglion has about 10 of these nerves, and that they fan out to all parts of the mantle wall. He also showed that each of these stellar nerves contained a large, transparent tubular structure, about 1 millimeter in diameter, which Young at first took to be a blood vessel. In transverse sections, though, he found that this structure was, apart from its size, little different from the numerous small nerve fibers which surrounded it. Moreover, unlike a small arteriole, it never contained blood or amoebocytes. Examination of the stellate ganglion, particularly in the small transparent squid Haloteuthis, showed that all these tubular structures arose within the ganglion itself. Closer examination of the dissected ganglion confirmed this, and detailed studies of its histology convinced Young in 1936 that each of these tubular structures must be a single nerve fiber, a giant axon formed by the fusion of very many smaller ones. Young confirmed this hypothesis when, using a pair of simple electrodes, he stimulated a stellar nerve. Large contractions of that part of the mantle served by the stellar nerve were only obtained if the tubular structure within it remained intact. These structures, therefore, must be single nerve axons, each almost 1 millimeter in diameter.
Okay, so those are ganglion. We talked about the ganglion. You saw the nerves coming out. What is a ganglion? Grouping of cell bodies, very good. And so what do you suppose these axons are doing? You saw what happened when he stimulated the axon. What are these axons doing in real life, do you think? Motor functions. They're moving the squid around, allowing it to swim, because when you stimulate it, you saw the contraction. So that's exactly, they're motor neurons. But they're huge. You can see them by eye. And so that allowed these guys at the first time. Now we have much more fine technology, but this is way back in the 1950s. They were able to first identify these large axons and then determine that they were neuronal axons and then record from them specifically. So let's just look at the next one quickly. We're going to look at part of this next movie. All of this was done at Cold Spring Harbor Labs right up in Massachusetts.
An electrode was first placed inside the axon by Hodgkin and Huxley in 1939 at Plymouth, and by Curtis and Cole in 1940 at Woods Hole. Hodgkin and Huxley first made a plastic cell and mounted it on a platform which could be raised and lowered like a lift. As Professor Baker shows, the axon held by the cannula was then hung in the cell, which was filled with seawater and connected to an external electrode. The internal electrode, which was not attached to the lift, was then placed vertically above the axon, with its tip in the cannula. It was centered with respect to the cannula and axon by placing a small mirror beside the axon, arranged so that a second image at right angles to the first was seen through the microscope. By adjusting the position of the cell, both horizontally and vertically, the cannula and axon were now raised up over the tip of the electrode, which always remained in the same line of sight. Hodgkin and Huxley found that as the electrode entered the axon, a negative potential with respect to the external seawater of about 65 millivolts was obtained. This was the resting potential of the axon, and although its existence had long been suspected, this was the first time it had been directly measured.
So that's exactly how they first showed definitively that the inside of the neuron at rest has this negative potential. And then all the rest that we went over, the proteins, the potassium, that was figured out in large part by Hodgkins and Huxley as well. Okay, let's get back to the lecture. Okay, so as I said, they measured for the first time the resting membrane potential in a living neuron from a giant squid axon. So now we move on to the second part. Now we figured out why there's a negative resting potential. But you don't want to, neurons are just not at rest. They do things. They fire action potentials. So now we're going to ask the question, how do you change this resting membrane potential, and what happens to all these ions and all these channels when you change it? So how do you change it? Well, inputs from other neurons can change the resting membrane potential of your target neuron. And input from presynaptic neurons can cause changes in electrical potential across the membrane of the postsynaptic neuron. Our postsynaptic neuron here is the neuron that we are interested in. Now, there's two types of changes that you can see in our target neuron. Hyperpolarization is an increase in the membrane potential where the interior of the membrane becomes even more negative. So I told you we started out at rest at negative 60. Hyperpolarization would be going down to negative 80, negative 100, negative 120. That's hyperpolarization of the neuron. In contrast, depolarization, as you might be able to guess, is a decrease in the membrane potential. The interior of the cell becomes less negative, going towards zero and sometimes even becoming positive relative to the outside. So you must know this. What is hyperpolarization of a neuron? Going more negative. What is depolarization? Going more positive. Okay? So that's what could happen. How that happens is through inputs coming in. But let's look at the differences between a hyperpolarization and a depolarization, because they do two very different things. A hyperpolarizing stimulus produces a response that passively follows the stimulus. So if it's a big stimulus, it'll be big, and then it'll disappear, and then the stimulus disappears. A small stimulus will be small, and as I just said, the greater the stimulus, the greater the response. And we say the change in the potential is called a graded response. Why is it graded? It's graded for a couple of different reasons. So here's my stimulation. I'm giving a little stimulation, hyperpolarized. I'm going more negative, little bigger, little bigger, little bigger. Here is my stimulation. Here I'm recording at two different spots. One I'm recording right here on the axon, and here I'm recording on that part of the axon. Here with a tiny little stimulation, I get a tiny little hyperpolarization that is measured right here on the axon if I'm stimulating right here. And a little bigger, I get a little bigger hyperpolarization, a little bigger, and a little bigger. But very interestingly, you'll see that from the same stimulation here, all of those signals are muted when you get even farther away. So now I'm recording down here. It's farther away from the stimulator, and I get very little response. So it's just like, if you're standing next to somebody, you shout in their ear, they get the message really, really loud. But if a second person is on the other side of the room, yeah, they might hear something, but it's very, very weak. So that is called a graded potential, okay? So it follows the strength of the original stimulation, but it really degrades as a function of distance away from the actual stimulation site, okay? And that's what we call local potential. That is, as the potential spreads across the membrane, it diminishes as it moves away from the point of stimulation. Okay, so that's hyperpolarizing stimulation. It's graded. The farther away you are, the less effective it is. It's not that great of a communication mode. But that differs from a depolarizing stimulus. Now, the depolarizing stimulus is the same as a hyperpolarizing stimulus to a point, but there's a special point where a flip is switched. A switch is flipped. If the membrane reaches a threshold, this is an important term to understand, a threshold of about negative 40. We started at negative 60. It has to depolarize to the special point of negative 40 millivolts. It doesn't just keep going higher and higher. It triggers what we call an action potential, which is a very, very brief burst of electrical activity. And during this brief, this temporally brief burst of depolarization, the membrane potential reverses and the inside of the cell becomes positive, but just for a few, very, very short time, okay? So there is a threshold, -40. Once you reach there, you get an action potential. Let's, okay. So as I said, action potentials are brief but large changes in the membrane potential. They originate in the axon hillock. Does anybody remember where the axon hillock was? Yes. Yeah, go ahead. Sorry.
Where the axon starts?
Right, just where the axon, right. So there's a cell body, and then there's the axon starting. Right where that axon starts, that's the axon hillock. That's a very, very important point, because all of these action potentials are originating in the axon hillock. It's like the sensory center of the neuron for sensing the potential and sensing whether the neuron has reached this critical threshold of -40. And once the axon hillock reaches -40, an action potential is generated. It is these action potentials, not just one, but the pattern of action potentials that carry information to postsynaptic targets. So this is, again, the mode of communication of the neuron. So we've gotten to the mode of communication. So let's focus on comparing and contrasting hyperpolarizing stimuli here from depolarizing stimuli right here, okay? So remember our hyperpolarizing stimuli. Here is the stimulation, little stimulus, little bigger, hyperpolarizing stimulus, bigger, bigger. We're recording from two different points, and we get this kind of increasing response as the increasing hyperpolarizing stimulation comes on. And as you get farther, you're farther away. Depolarizing stimuli, it's going up. You're stimulating to a positive, more positive potential here. And what happens is you get a little bit of now depolarization until you reach this dotted line, which is the threshold of -40. Once the cell's membrane reaches this, you gave it this amount of stimulation to make the membrane potential go, then you get this huge action potential that's going way above 0 and coming back down to what's called the after potential, and then it comes back to the resting potential. This brief increase and depolarization is the action potential. It's also called a spike. Okay, so how do you think about these action potentials and the generation of the action potentials? They have some very unique properties that you need to understand. One is it's an all or none property of action potentials. The neuron fires at full amplitude or not at all. It's not graded like this response here. You know, before it gets to the action potential, you could get a little bit of a graded response. But an action potential is all or none. How do you think about this? What is a good analogy? A good analogy that is very memorable is something that you're all familiar with. And I like to call it the flush analogy of the action potential. So you could flush a little bit, and you get a little bit of water flow. But a flush on a toilet is really all or none, right? You flush it, and it goes all or none. And you can't go back, and you can't reflush for a little while. That's a refractory period. We're going to talk about refractory periods. It is an all or none. These action potentials, like a toilet flush, are an all or none event. Action potentials increase in frequency with increased stimulus strength. To a certain degree, they can't come too frequently, and we'll talk about why that can't be. And after potentials are changes in membrane potential after the action potential. And here is a hyperpolarization. It goes lower than the resting potential. That's the after polarization before it comes back to reach the resting potential again. Okay, why do you get these action potentials? What is causing this increase in in the electrical environment of the cell, what's happening. And that is caused by the movement of sodium ions. And so now those sodium ions that were waiting to get in, they were outside, couldn't get in because there's no ion channels open, they are the ones that flow in to the neuron to cause a brief but huge increase in the potential of the inside of the cell. At the peak of the concentration gradient, pushing sodium ions in equals the positive charge driving them out. And the membrane shifts, the inside shifts briefly from a resting state to an active state and back. That is, it goes from the resting state, -60, to a very positive state, about plus 40, and back to the resting state. And so it was also Hodgkin and Huxley that, that showed the flow of sodium ions was critical for the production of the action potentials. We're just going to jump back. Oops, I forgot to take that out. Jump back to the movie because it continues right here.
Moreover, when the axon was stimulated, the action potential did not simply fall to 0 during the impulse, but became positive with respect to the outside, shown by the overshoot of the action potential.
Here's the action potential.
This important
plus
discovery
40.
suggested that the nerve membrane, which at rest is mainly permeable to potassium, becomes primarily permeable to another ion during excitation. This other ion is sodium, since if its concentration in the external solution was lowered, the action potential immediately became smaller, by an amount depending on the sodium concentration. If, as these experiments suggest, the action potential was dependent on the passage of ions across the membrane, it was obviously important to measure the currents carried by these ions. To do this, it is necessary to hold the internal potential at a chosen value. This is the powerful voltage clamp technique, originally developed by Cole in America and applied by Hodgkin, Huxley, and Katz. This technique requires that an extra electrode, the current electrode, be inserted into the axon. For this purpose, a double electrode was made by winding very fine silver wires around a thin glass capillary. As the wire, which was only 20 microns in diameter, was wound on, it was kept taut, as Sir Alan Hodgkin shows, by dangling a small piece of plasticine on the free end. The finished electrode consists of 2 entirely separate spirals, insulated where necessary by shellac varnish. While it is inserted in the same way as a simple electrode, the information it gives is quite different. As a change of potential is imposed on the axon, as seen in the top trace, the currents flowing across the membrane are revealed in the bottom trace. The early downward dip, seen on the left, is the transient current carried by the influx of sodium ions, and it is superseded by an opposite and persistent current attributed to the outward flow of potassium ions.
Okay, so now we know that what you can do to determine whether it is potassium or sodium that's changing, that's causing the action potential. All you do is change the concentration. You don't let any potassium ions you don't let any sodium ions in the extracellular space, or you decrease them, and you see a very decreased action potential itself that showed definitively that it is the sodium ions flowing into the cell causing the action potential. So let's go back to our current understanding of these events. So what Hodgkin and Huxley showed, and this is very, very simplified. They had a really amazing set of findings for their Nobel Prize. Among other things, they showed that action potentials were created by the movement of sodium ions into the cell through channels in the membrane. And they modeled these changes in potential with elegant mathematical equations as well. We're not going to go over that. But we do need to talk about how the sodium gets into the cell when the membrane reaches this critical threshold. What's so special about the threshold? So now we're going to step back and remember what we talked about at the beginning, the different kinds of ion channels. And we're going to talk about the voltage-gated ion channels, okay? So voltage-gated, this time, sodium channels are our key target for the membrane. We're going to add yet another kind of channel in our neuron cell membrane. Right now we only had the sodium-potassium pump, and we had the potassium channel open all the time. Now we're going to add to our membrane the voltage-gated sodium channel. This channel is open in response to the initial depolarization. That is, it's open when the neuron gets to its threshold of negative 40 millivolts. The more voltage-gated channels open, the more sodium enter the more sodium ions enter the cell, and this continues until the membrane potential reaches the sodium equilibrium potential of plus 40 millivolts, and it's again the Nernst equation that will tell you what the sodium equilibrium potential is. Okay, so. The second voltage-gated channel that we're going to talk about, so we have voltage-gated sodium channels. Now, in addition to the vanilla regular potassium channels open all the time, those are still open all the time, we have now a voltage-gated potassium channel. And this is like an extra safety valve. As the inside of the cell becomes more and more positive, it actually activates the voltage-gated potassium channels. And what happens is the inside is becoming very, very positive. There's too much positivity in there. And so as these voltage-gated potassium channels open, it allows even more of an exit gate for potassium to move out. And the resting membrane potential is restored. But now you have this inrush of sodium and outrush of potassium. We're now not quite at resting potential. Now is where you see the value, I hope, of the sodium-potassium pump. It's after these action potentials, when you're out of balance and you have more potassium inside the cell more sodium inside the cell and potassium that has just gone out. Now you see the value of this energy-deriving pump or energy-using pump that pumps out 3 sodium ions for every 2 potassium ions coming in. Now you can see why the cell needs that, okay? Any questions so far? Is that clear? Okay, good. So let's go through this step, and I would highly recommend, this is the figure from your book. You really need to understand each one of these figures and understand each one of these schematic diagrams that we're going to go over. Very, very valuable. Okay, so we're going to start at rest. Resting, we started with this at the beginning of the lecture. We have our vanilla kind of potassium channels open all the time, just open all the time. But we have 2 more channels that I just told you about. These blue ones are voltage-gated sodium channels, and this shorter one, brown one, is the voltage-gated potassium channel. The only one open for business all the time is the regular vanilla potassium channel. We're at rest. We have negative 60 millivolts on the inside relative to the outside. But forces come in, inputs come in, and the membrane is depolarized, and it hits this negative 40 millivolts, the threshold for the action potential. And what happens is you get, once you hit the threshold, we jump to here, and you get immediate opening of these voltage-gated sodium channels, okay? Voltage-gated sodium channels open just as the membrane potential hits -40, and what happens is, remember, you have all those sodium ions waiting to get in. There's no sodium on the inside, and so those sodium ions, once that channel is open, they rush in. And that causes that huge and fast increase of the membrane potential up and up and up. The more the membrane potential goes up, the more sodium ions go up until it reaches plus 40 millivolts. Again, that's the resting potential there, and then there's no more rush of sodium ions in. Because sodium is coming in, it's getting very, very positive, you have the potassium ions fleeing out because it's becoming too positive inside. That happens. But in addition, what you have is a little bit later, after the potential has gotten really, really high, you get the activation of the potassium, the opening of the voltage-gated potassium channels. At the same time, the opening of the voltage-gated potassium channels helps even more potassium come out of the cell. And at the same time that these potassium channels, voltage-gated potassium channels are open, you get an inactivation of the sodium channels. So these kind of get worn out. And so they open up, they rush, they rush in they allow all of the sodium to rush in, and then they get inactivated, okay? And then finally, when the membrane comes back down, of course, these just close because they're not activated by the change in the membrane potential anymore, and the voltage-gated potassium channel, again, they're only activated way up here at this voltage. They close as well, and you're left with a situation kind of like this, but now you have more potassium on the inside, more sodium on the inside and less potassium on the inside, and this is where your sodium-potassium pump starts to work to bring everything back to equilibrium. Okay. Now, I also want to go over two very important concepts. The major concept is called refractory period. This is a period where it's much harder to elicit a second action potential after a first action potential has been elicited. There is two different kinds of refractory periods, an absolute refractory period and a relative refractory period. The absolute refractory period happens in this relatively short period right here. In this time, there's no way you can do like a double action potential here. It's everything, all your sodium channels are being used for the single action potential. No more sodium channels to be activated to get like a double potential. This is absolute refractory period. No possibility for another action potential. This is the relative refractory period. Here, you can possibly get a second action potential, but you have to really stimulate it. And you have to really stimulate it because at this point, you have your voltage-gated potassium channels opening up. They're letting more and more potassium channels out. You're getting more negativity on the inside. So you really have to stimulate hard to activate all of the possible sodium channels that haven't been deactivated from the first action potential. It can happen, but it's very hard, and then we call that the relative refractory period. And then in this point, when the membrane potential is getting back to resting, then it goes back to its normal state. Okay? So I just wanted to remind you of these factors underlying the resting membrane potential that we talked about. The negative intracellular proteins, the sodium-potassium pump that is mainly now, you understand, active after action potentials to rebalance the things coming in. Sorry, yes?
What's the time frame for the action potential?
Yeah, so it's very, very short. It's on the order of milliseconds. So action potentials can't fire more than one every couple of milliseconds. So this is a very short, very short time frame. Good question. Yes?
What factors cause the initial reaching of the threshold point?
Yeah, so the factors that cause the reaching of the threshold are, let me just go back. So the question is, what are the factors that cause the membrane potential to change? And here, it is the inputs from the other axon onto this cell. And that's the chemical transmission that we're going to go over next time. So we're going to talk about that. But just know that it's the inputs from the axons terminating on the dendrites and the cell body of this neuron that are changing the potential. It could change up or down. There are inhibitory inputs as well, but we're focused on excitatory inputs. Any other questions? Okay, good. Great. Okay, so you now understand the importance of the sodium-potassium pump to get everything back to balance. At rest and during the whole action potential, it is permeable to potassium. During rest, it's not permeable to sodium, but now we told you that there's a voltage-gated sodium channel critical for the action potential. And still, these two forces are still at working during the action potential, the force of diffusion and the force of the electrostatic forces, okay? So, and then these are just definitions of refractory period. Only some stimuli can produce an action potential. Absolute refractory period, no action potentials are produced. That's a very, very narrow time period where there's an absolute refractory phase. And relative refractory phase, as I said, only strong stimulation can produce an action potential. And by strong stimulation, I mean lots and lots of inputs from other neurons. Okay, so concepts from this first lecture. And please ask your questions now while it's all fresh, because we have just a few minutes. Electrical versus chemical transmission. Of course, we focused on electrical transmission. We will cover chemical transmission on Wednesday. Yes? Does what happen? At every node. So action potentials by node, do you, so she asked, oh, node of Ranvier. So do you mean the axon hillock? Yeah. Yeah, that's a good point. So we're going to talk about the node of Ranvier is the part of the axon that is not covered by myelin. And what happens is the action potentials jump from the hillock to the first node of Ranvier to the second node of Ranvier, and you get this whole sequence that I talked to you about, the rushing in of potassium and the rushing out of the rushing in of sodium, the rushing out of potassium, just at that node of Ranvier, and then it jumps to the next node of Ranvier. And that is exactly why the myelin speeds conduction, because if you don't have the conduction, you do have action potentials happening every little segment of the neuron that you can imagine slows things down. So yes, that gets a little bit ahead what we're going to talk about. We're going to talk about that at the beginning of the next lecture. Good question. Any other questions? Yes? What's happening inside the neuron when there's a negative?
Sorry, what's the purpose of the charge change? If there's an impulse being sent, stimulus that changes the charge?
Yes.
What does the stimulus actually do? Is it sending some kind of particular message? Does the message change?
Okay, so is your question, what is the nature of the action potential message? Okay, so what is the nature of the action potential message? And that is actually a very deep, interesting question. There are at least two modes of thinking about that. Some think that you can simply average the number of action potentials and say, like, if I have neurons that respond to your face, and certain neurons are face-selective neurons, and I have a huge response to your face, but I have a very little response to your face, for example. That is the nature of the signal. You can provide information about selectivity to particular things just by kind of the volume of action potential output, okay? That's one idea. So big means I'm highly sensitive, little means I don't care about you. Others say that that's too crude for our neurons that have been evolved in this very, very precise way. And they say that it's not just the volume of action potentials that this neuron is firing. It's actually the pattern of the action potentials. So kind of like a Morse code. So is it a Morse code of if I have a particular pattern that says something, and I have a particular pattern to you, and I have a different pattern to you, and I have a different pattern to you, that might be a richer way to convey information. And that is a very, very deep and complicated debate. But that is basically what it's conveying. Information about whatever system that you happen to be in. I gave an example of a face selective system. Was that the question?
Yeah, but then I'm also wondering what a stimulus would be that would, what hyperpolarizing versus a depolarizing
Okay, right. So hyperpolarizing stimulus will just not allow that neuron to fire. So let's say I'm a neuron and I really like to fire to your face. But I have another negative neuron, a GABAergic interneuron that is firing at me and does not let me fire at all to your face. I become insensitive to your face. This neuron simply cannot fire. And it's a push and pull. Maybe I don't want to fire to your face every single time. Maybe I have to ignore your face because I have to pay attention to other faces. But I'm a neuron that likes your face. How do I ignore your face? I'm just a neuron and I like to ignore your face. Well, I have negative inputs on me that suppress my firing to your face. And you can imagine that is kind of executive control. That is filtering out at a cognitive level certain firing patterns when I don't need to pay attention to your face and I have to look for somebody else's face, for example. Okay? Great. So just in the last 2 minutes, be familiar with all of these terms. There are really important exercise questions that you will, questions for this lecture. Make sure you can answer those. And again, I recommend that you read the book, review the book for the chemical transmission lecture on Wednesday. Okay, see you all Wednesday.